Okay, so you want to learn about photosynthesis, but you don't want to do your own research, and you don't want to watch somebody else's video because you might accidentally learn too much. Then you've come to the right place. So I'm going to give you an AP Bio rundown on photosynthesis, and it all begins, as this diagram shows, with the sun providing light to our plant here. That's a flower. And the plant is taking in carbon dioxide and also water from its environment, and it's releasing oxygen, but it's also creating glucose, which is not shown in this picture. Now the carbon dioxide and oxygen exchange occurs in little ports here on the underside of our leaves called stomata, and stomata open and close to allow the carbon dioxide in and out. Generally they're open at night, just because during the day it's a risk of losing too much water in the heat of the sun. The water is actually traveling up through what's called the xylem, and that transpires out through the stomata, and that helps to continue drawing water and other nutrients up from the roots. Meanwhile, there's also tubes called the phloem, which bring the glucose back on down to the parts of the plants that don't have access to glucose and other carbohydrates just because they're deep underground and they can't do photosynthesis. This is a view of the process of photosynthesis. So we split it into two equal halves. We have the light-dependent reactions, and then we have what are called the light-independent reactions, or called the Calvin cycle, or sometimes called the dark reactions. As the name implies, those reactions don't require sunlight directly, but they require energy-carrying molecules from the light-dependent side. So looking at the left-hand side of this, you see the light coming in, you can see water coming in right here. That's coming into these components of the chloroplast, which are called thylakoids, and a stack of thylakoids is called a granum. In truth, the chloroplast is like chock full of these things. They'd be all over the place. And inside of the thylakoid is the chlorophyll, which makes the thylakoid appear so green, and the fact that we have so many of these stacks, these granum, throughout the chloroplast is the reason why the chloroplast is so green. And then typically plant cells that are part of the leaves and stems of the plant are chock full of chloroplast, which is why the whole plant then seems green. Diving into this a little bit more closely, here's just the same picture, but it's much more colorful. So if you want to pause and just take a look at this, it's much more kid-friendly. But diving a little bit more into this, with the light-dependent side, we have a number of reactions happening inside of the membrane of the thylakoid. So what happens is light's coming in, and that light's being captured at two different points. We call them Photosystem 1 and Photosystem 2. Now, just to throw you off and confuse you, Photosystem 2 kind of comes first in most of our diagrams. It's not so much that it happens first, but really they're happening at the same time. But Photosystem 2 is the one that I'm going to start with this explanation, because we have light here coming in, and that light is going to excite an electron. So there's an electron here, and he's just hanging out, being an electron. Pretty boring life. And all of a sudden he's got all this energy from these photons coming in from this light. And the photons that are being captured most predominantly in Photosystem 2 are of wavelength 680 nanometers. That's only really significant to compare against Photosystem 1, which is capturing slightly different wavelength light. The excited electron then is liberated from that chlorophyll, from that photosystem, and it's able to go off to other places within that membrane. And there are proteins which can scoop up that electron and basically use the energy of that excited electron in order to get other things done. And one of those protons is pictured here, and this is a proton pump, which is moving hydrogen ions, positive hydrogen ions, into the thylakoid. Now, a positive hydrogen ion is just a proton, right? Because a hydrogen is a proton and an electron. So if you have a missing electron and it's a positive hydrogen, it's just a proton. So those protons are being pumped into the thylakoid. That electron now is no longer excited, and it ends up over here at Photosystem 1. Now that works out because at the same time that Photosystem 1 is receiving an electron, it's having its own electrons excited by light again, and that excited electron is then zooming off and doing other things. So it's got one out, one in. So Photosystem 1's pretty happy about that. Now the electron that is leaving Photosystem 1 actually goes on to reduce NADP into NADPH. So we have a reduction reaction happening here, and we end up with NADPH, which is one of our energy-carrying molecules that's going to power the light-independent side of photosynthesis. Now, I mentioned that photosystem 1's pretty happy with its 1 electron in, 1 electron out thing. So if we go back to photosystem 2, photosystem 2 unfortunately was losing an electron, but it doesn't have another electron like coming in from another photosystem. That doesn't exist. And so what happens is it gets this deficiency of electrons. And when that's significant enough, that's going to encourage water molecules to split. And when they split, they're going to basically donate back an electron. So that photosystem 2, for each electron that is being excited and lost, it's going to gain an electron from the splitting of water molecules. And each water molecule that is split is going to liberate 2 hydrogen plus ions and donate 2 electrons to that photosystem 2. Now, if you're keeping track here, we've got a proton pump pumping in hydrogen ions, and here we have additional hydrogen ions. We're getting this huge concentration of hydrogen ions inside of the thylakoid, and diffusion's going to be pushing those hydrogens to leave. Right, so we have this huge high concentration. They want to get out of the thylakoid, but they don't really have a way through the phospholipid bilayer. Luckily, there's this protein over here called ATP synthase. And what ATP synthase does is it provides a little channel for hydrogen ions to leave the thylakoid so they can flow on out of here. But ATP synthase is kind of like, it's like a little water wheel or like a revolving door here. It's not physically spinning, but it's capturing the energy of these hydrogen ions pushing through, wanting to leave the thylakoid. And it captures some of that energy and it uses it to take ADP and recharge it into ATP, triphosphate, right? So it adds that phosphate group back in. And now we have 2 energy-carrying molecules, NADPH and ATP. The reason why that's significant is if we jump back to our diagram here, we can see that the light reactions, which are receiving the light, are producing those 2 molecules, and those 2 energy-carrying molecules are then what's going to go into the Calvin cycle to power that. So just from a different view here, we see light-dependent side takes in the light, charges up ATP and NADPH, which can then go off to the Calvin cycle. Taking a closer look at the Calvin cycle, at first it looks completely crazy. You've got all sorts of molecules going on. It's hard with any cycle in biology to really pick a place to start. I'm going to pick right here the input of CO2 into this cycle. So we have 3 CO2 molecules coming in. They're going to join up. Each one is going to join up with a 5-carbon molecule called ribulose biphosphate. And ribulose biphosphate has 5 carbons. So each one of those 3 molecules scoops up a CO2 and it becomes a 6-carbon molecule very temporarily, because that 6-carbon molecule is actually very unstable, so it almost immediately breaks into 2 3-carbon molecules. So all 3 of these scoop up an extra carbon and become these 6-carbon molecules, and then they break in half. So now we have 6 3-carbon molecules called 3PG. The 3PG is going to go through process requiring ATP, requiring NADPH. It's changing form, and it eventually becomes this guy down here called G3P. Now the exact names and exact structure of all of these things is kind of outside the scope of what we need to worry about right now, but understanding that process of this 6-carbon molecule that's unstable breaks into 3-carbon molecule, and then we need energy to drive the changing and building of that molecule into G3P. That's significant. And the reason why that's significant is because this G3P down here is going to become the part of the glucose molecule. So one of these 6 G3Ps is going to leave the cycle each time we go around. So that's 3 carbons coming out. So every 2 times we go through this cycle is going to yield us a glucose molecule. The other 5 G3Ps then continue on, and through additional processes which require more energy, they're going to restore the ribulose biphosphate. And then the cycle can continue, scoop up more CO2, turn into 3PG, go into G3P, and then spit out one of these molecules each time. And that's the key to what's going on here. Every 2 of those you get becomes a glucose. And as we get more and more glucose, those can develop into starches or cellulose, these larger molecules that are the structural and energy storing components of plants. So the vast majority of what a plant actually is, when you look at it, is made out of these glucoses that have been built through the process of photosynthesis, which is why from almost the very beginning, as this seedling sprouts out of the ground, a plant's going to require regular access to sunlight and CO2 and water in order to grow. If we go back here, so just a quick recap of what's going on here. We have sunlight being captured by the physical components of our plant. The light dependent side is capturing that energy and storing it up in our energy carrying molecules ATP and NADPH. Those then get handed off to the Calvin cycle, where they supply the energy for creating glucose from CO2. The NADPH and ATP, once they're depleted of their energy, return to the light dependent side as ADP and NADP, and you can see that right here. Here's the NADP, and the ADP is here as well. So those molecules are coming back in, getting charged up again, and then they go back off to the Calvin cycle, and it's of course its own cycle. That is the process by which we take light, we capture it, and we turn it into something that's a little bit more useful. Plants use the sunlight in order to produce the glucose. They can't just use sunlight, because then their cell membrane and all their different cell parts would need like direct access to the sun and a way to turn the energy of those photons into like the ability to do the work that they need to do. Which of course is complicated and it would be really convoluted and weird. This whole process and all these many steps and all these enzymes and all these different parts that need to work together is all about taking sunlight and turning it into something that's a little bit more easily used, which is that glucose. But of course we know that glucose itself is not actually that readily available and easy to turn into instantaneous energy, which is why we need cell respiration, which, spoiler alert, takes our glucose and turns it into tons and tons of ATP, which is actually like very easily and readily usable. That's for another day. So this is photosynthesis. Hope you had fun.